Glycolysis. Next slide. Glycolysis introduction. Glycolysis is derived from Greek word glucose lysis breakdown. During this process, one molecule of glucose is divided into two molecules of pyramide. Free energy is released in this process and is stored as two molecules of ATP and two molecules of NADH. History. Glycolysis was the very first biochemistry studied, and it is first metabolic pathway disorder. Lucy's pleasure 1854-1864 observed that fermentation is caused by microorganisms and also found that aerobic growth required less glucose than anaerobic conditions. Third, carbohydrate metabolism. There is 60 of ingested food consists of polycystic carbohydrates, which while the discharged carbohydrates like sucrose and lactose are important in the diet of infants. The end product of carbohydrate digestion is mainly glucose, fructose, and lactose. These simple sugars are absorbed in the intestine and are carried to liver through the bloodstreams. Here they are transferred into glucagon and glucose 6-phosphate. The glucagon is stored in the liver. The carbohydrate metabolism or design of carbohydrate is taken to the two types: catabolism and anabolism. Catabolism. In catabolism, degrades of complex organs molecules into simple molecules. In this process, energy is released. Anabolism. Anabolism is the process biosynthesis which complex molecules are produced from simpler molecules. In this process, energy is required. Glycolysis is the breakdown of glucose up to the fermentation of fulvic acid. Each glucose molecule forms two molecules of pyruvic acid. The breakdown of glucose is taken place in the series of steps such as this reaction of catalyzed by the specific enzyme. Glycolysis may be divided into phase: perforate phase, oxidative phase. Perforate phase. The first 1 to 4 steps in glycolysis represent the perforate phase. In this phase, breakdown of glucose and low energy phosphate occurs in the first is expanded. Oxidative phase. The end step in the glycolysis represent the oxidative phase. In this phase, high energy phosphate bonds are formed and energy is stored. Phosphorylation. Glucose is converted to ATP into ADP. Glycolysis 6-phosphate forms the fructose 6-phosphate. Then it converted into ATP to ADP. Fructose 1,2,6 diphosphate. Then it will go to the isomerization. Pyruvic acid, the left is lactic acid. Lactic acid fermentation, alcoholic fermentation, acetylate which leads to ethyl alcohol. Steps involved in glycolysis. Phosphorylation. It is the first step in the breakdown of carbohydrate. The glucose is stable compound, so it normally releases the breakdown. The activation of glucose molecules take place by a reaction called oxidative phosphate. The phosphate group is attached to the glucose by a low energy phosphate bond, and glucose 6 phosphate is formed. The reaction is facilitated by the enzyme called hexokinase with Mg2 plus as an activator. The phosphate group is derived from the ATP which broke down isomerization. Glucose 6 phosphate undergoes internal molecules rearrangement to form fructose 6 phosphate. This catalyzed enzyme is phosphoglucosaminosis. No changes take place in the low energy value of phosphate bond. Second, phosphorylation. Fructose 6 phosphate undergoes phosphorylation to form fructose 1 6 diphosphate. The catalyzed enzyme is phosphofructoglycanase. The phosphate group is divided from ATP which break down the ADP. Cleavage. Fructose 1 and 6 diphosphate splits into two halves between carbon atom 3 and 4 under the action of enzyme aldolase. The two halves phosphate each contain the 3 carbon atoms but not identically. The one half of diractoserance phosphate DHAP and the other 3 phosphoglycosaminases. These molecules under isomerization and become identical 3 phosphate PGAL molecules. The catalyzed enzyme is triphosphate isolation. அடுத்தது பேரு என்னடா இது வந்துகிட்டே இருக்கு 3 ஸ்லைடு இருக்கா ஒரு தடவை வாஸ்து பார்த்துட்டு அடுத்து வந்திருக்கணும் சரி ரெடியா 6 ATP generation 1 2 3 6 ATP generation 1 and 3 diphosphoglycerine acid now transfer into phosphate which high energy bond to ADP 3 phosphoglycerine acid. The catalytic enzyme is phosphoglycosylate. ADP acquires the high energy bond phosphate and becomes ATP. Isomerization. The 3 phosphoglycerate acid molecules undergoes initial rearrangement and become 2 phosphoglycerate acid. The catalyzed energy is phosphoglycerization molecule. 8th one is the 2 phosphoglycerate acid molecules lose hydrogen and oxygen in the form of water to form phospholane pyruvic acid. The step is catalyzed by the enzyme phenazoles. ATP generation. The என்ன போட்டிருக்குது ATP generation. The phosphoenol pyruvic acid molecules transfers its high energy phosphate bond ADP which converts into ATP and pyruvic acid is formed. The reaction is catalyzed by the enzyme called pyruvic kinases. It should be not that 2 molecules of pyruvic acid are formed per molecule of glucose metabolism. For biological signification. In the absence of oxygen, pyruvic acid is converted into either ethyl alcohol or lactic acid. Pyruvic and lactic acid may return to the liver where they can be resynthesized to form glucose or glucagon by release anaerobic glycolysis. Lactic and pyruvic acids may also be broken down in the liver to yield carbon dioxide and water through Krebs cycle. Thank you. அவ்வளவுதானா